Wait, does that mean I start or? Oh, 1 more minute. Okay. All right, I'll start now then. All right, hi everybody, thanks for coming to my talk today. Today I'll be talking about adaptive radiations. All right, so adaptive radiations is basically the process where from a single common ancestor, you see this rapid diversification of many different lineages that have diversified into these various phenotypes and ecological diversification. Now, because of this remarkable diversity, this phenomena of adaptive radiation has really attracted the interest of many evolution and ecological researchers, and we really wonder how has this evolutionary process has occurred. Now, for me, I'm really interested in about adaptive radiations because there seems to be a little bit of a paradox on how this can happen, especially in island settings. Because when normally islands gets colonized, you go through this process where there's this population bottleneck that happens here. And what usually happens with bottlenecks is you lose all these genetic variations. So from this loss of genetic variations that you're able to then have this rapid diversification of many different lineages, almost seems like this is a biological paradox that is happening. So I'm really interested in how this potential biological paradox happens in nature. And the study system I study is a group of tree species called the Metrosideros. Now, Metrosideros is a group of plants that consists of 60 different species, and many of these, and it's a woody plant species where its forms can be found as trees, shrubs, and vine plants. And they're widely distributed across the Pacific basins and many different Pacific islands. Now, they're animal pollinated outcrossers and they're wind dispersed. And this is especially an important plant species that are found in several different island forest ecosystems, and especially it's important is in Hawaii. Now, the most interesting thing about the Hawaiian Metrosideros is that this is a case of an adaptive radiation where there are more than 25 different taxa that are found across the Hawaiian archipelago. And what's really noticeable about this is that these are morphologically diverse, but they're also ecologically diverse as well. Now, what makes this Hawaiian adaptive radiation really interesting is that if you look at the geographic route of reaching Hawaii, the ancestral species of Metrosideros is thought to have arose from New Zealand, and then it hopped onto multiple different islands to reach Hawaii. And if you think about the number of constant bottleneck after bottleneck it had to undergo to reach Hawaii, and then from reaching there, it all of a sudden went into adaptive radiation, really looks like a fascinating evolutionary phenomenon. So we're really interested in understanding the basis of this adaptive radiation. So what we decided to do was take a genomic study to this. And so first what we did was we decided to assemble a very high quality reference genome for the Hawaiian Metrosideros. And then we decided to sequence over 130 different individuals that spans 11 different taxa that I'm just, and these letters represents the name of the different taxa that I've just abbreviated into single letter alphabets. And basically, whole genome sequenced it to a pretty high depth of a 14x, and we basically analyzed over 22 million SNPs. Now, basically taking this data, what we initially did was we did a phylogenomic analysis, and this is the initial phylogenomic tree that I'm showing you. And let me take you step by step on what you're seeing. First, what you can see is that individuals are largely grouping by their island of origin, and within the Oahu Island here, the taxa can be divided into this glabrous group and this pubescent group. And the glabrous and pubescent is how hairy their leaves are. Second is you can see that individuals are largely grouping by the taxon status as well. So taxons have been normally designated based on their leaf morphology, and as you can see on how hairy their leaves are. So the taxons classification makes sense on the genomic level as well. Now, while it seems like everybody is grouping by taxon status and island status, there are some groups that doesn't seem to make sense in terms of its grouping. For instance, the wild pubescent groups are grouping together here, but the wild glabrous groups aren't really grouping well, as you can see here. There's even weak bootstrap support, and some seems to be even grouping with the Hawaii Island Metrosideros. So we really wanted to understand what's going on here. And we decided to take the approach Twist takes, where basically you divide the whole genome up into 10,000 base pair windows, and each window you basically look at the topology of that window. And for the topology, we just focused on the 4 wild glabrous taxa here, and for 4 taxon case, you can have 15 different topologies. And what the results show is that there is no single topology that's dominating across the genomes, meaning that there's a high degree of this incomplete lineage sorting happening. Now, we really were interested and curious on what's driving this high degree of ILS, and decided to do a simulation study. Nowadays, everybody's using SLIM to do simulations, so we decided to do that as well. So we use SLIM to do the simulations. Some of the demographic parameters that we were interested in were estimated using DADI, and then we have this demographic scenario that we were interested in. One simulation that we were really interested in was there something interesting going on in the ancestral population size that have led to the incomplete lineage, a high degree of ILS, and simulated 3 different ancestral population size scenarios. And you could clearly see from this Twist results that when the ancestor had this large population size, you can see that there's this high degree of ILS, whereas compared to the small ancestral population size. So some of this is perhaps not too surprising for this crowd, but we're basically validated this with our simulation results. Now, the really interesting part about this is how does an island population have this high ancestral population size? Because didn't I tell you that going through this colonization events, you go through these bottlenecks? So why would this Hawaii population have a high ancestral population size? Well, one possible model is it's just because it happens to be like that. There was an ancestor that was really genetically diverse that colonized Hawaii Islands. Now, and we call this the single colonization from one genetically diverse ancestor. Now, we really don't think this is potentially the case because Hawaii is so distant from its nearby island where Metrosideros is found, which is in the Marquesa Islands. And it's more than 3,000 kilometers of open ocean. And in the Marquesa Islands, there's only one Metrosideros species, and that Metrosideros species has not undergone any evidence of adaptive radiation. So we don't think that's the case. What are the 2 alternative models then? Well, the 2 alternative models, what we think is basically the ancestor is just genetically diverse through some type of process where in this model, it's diverse because there's been a hybridization between 2 different species, whereas in this model, there's 2 genetically diverse species that colonize Hawaii Islands. So hence the single colonization or the multiple colonization model. How can we test which of the model led to the Hawaiian Metrosideros? Well, one analysis we did was looking at the past effective population size changes using the MSMC method. And what we see is that across the 11 different Hawaiian taxa, you see that there's this common shared bottleneck that has occurred 3 million years ago. And 3 million years ago was a time point when the oldest island of Hawaii, the Kauai Island, was started to have already formed and was available for colonization. So basically, this suggests that the Hawaiian Metrosideros had potentially single origin, meaning that the colonization model for the Hawaiian Metrosideros involves some type of single colonization with potentially a hybrid ancestor-like case that explains the high level of ancestral diversity. Now, lastly, what we were interested in was, well, what is the genetic basis of this adaptive radiation? Well, there's been multiple different models to explain how adaptive radiation actually happens. And one model championed by folks like Schluter and Simpson It's this ecological speciation model where from a uniform population, there's this divergence in environment that happens. And this population, as it adapts to these two contrasting environments, you see alleles that gets fixed to adapt to these two different environments. So this is basically the ecological speciation model, where a divergence in ecology leads to divergence in the genetics as well. So how do we look for this evidence of ecological the alleles associated with ecological speciation? Well, one way to do this is to use these FST scans, but specifically looking at taxa, where the comparison of the taxa in this case are taxa that inhabit very contrasting environments. So this is just a bunch of FST scans where we compared taxa that inhabit contrasting environments, and these yellow points here, that's a little bit hard to see, but I've highlighted here, are basically the outlier windows and the potential regions that are involved in the ecological speciation process. Now, we were really interested in what are these FST outlier regions that are potentially involved in ecological speciation. So we did, a bunch of Go analysis and all these things that I'm not going to be talking about today. What we were really interested in was what was the evolutionary origin or the selection process that led to this, the divergence in these SNPs for these regions. So basically asking is essentially, what is the evolutionary origin of the FST outliers? So how do we know the evolutionary origins of these FST outliers? Well, we can go back to the SNP region where there's a high FST, or there's basically the fixation of two different alleles between these two taxa that dwell in contrasting environments here. And then we can ask what is the evolutionary source of these alleles, by basically looking at the allele status of this in the other islands. So in model 1, where basically this is the model where there's been selection on a de novo variant, it's because this allele 2 that exists only in the Hawaiian riparian population does not seem to exist in any of the populations outside of the Hawaii Islands. So we can hypothesize that this is basically a de novo variant that arose. Now, the other model, of course, is that this allele exists in other Hawaii Islands, in the Kauai or the Oahu Islands. And in this scenario, it suggests that there's been some type of selection on ancestral variant. So with this in mind, we decided to basically do was first take all the SNPs across the Hawaii Island two taxa that dwell in the two divergent environments, look at how many of them are de novo, ancestral, and then we started counting how many of them within the FST outlier regions are de novo and ancestral, and we found that there's a higher proportion of those FST outlier SNPs that are within the ancestral class than the de novo class, and this difference is significant. So meaning that ancestral variants were more likely to be selected for forming these FST outlier regions. So at the end of the day, we come up with this model here to explain the evolutionary origin of the Hawaiian Metrosideros and potentially explain how adaptive radiations can happen in islands where there's these bottlenecks happens. The model basically is that there could have been these adaptable alleles that exist in multiple different Hawaiian species outside of Hawaii that came together through some type of hybridization process, and this hybrid this somewhat hybrid species then colonized Hawaii, and these adaptive alleles basically potentially gets reused as it's colonized the Hawaii environment, and then it becomes the fuel for driving the adaptive radiation. Now, the really interesting part of this model is that now we have a sort of a model that we can actually test in the future and start asking, well, what are these adaptive alleles, right? And what are these different species that are hybridizing, that hybridize to lead to the Hawaii population? And if this is the origin of the initial adaptive radiation, do subsequent adaptive radiation also means that this hybridization happened again? So there are these multiple models that we can test, and this is something that I would really like to test in the future. So with that, I would like to give acknowledgments to my postdoc advisor, Michael, also my collaborator, Elizabeth Stacy at UNLV, and also my funding sources, and also Evolution for giving me the supplemental grant, for the childcare supplemental grant for helping me getting here, and also the organizers for giving me this opportunity to talk. Lastly, I just want to bring a quick ad that I'm starting my own group at the University of Kansas, and this is something that you're interested in. And if you have friends that are interested in this type of work, please let me know. If you think this was boring, but if you find somebody who is interested in doing genomics in the University of Kansas, we have a postdoc fellowship that's really, really a good fellowship. So please get this word out. And if there's any other questions, I'll take questions. Thank you. Yeah, so there is a lot of hybridization happening, and for the ancestral variation model can be explained by hybridization of exchanging between alleles, right? So we've excluded that because it doesn't seem like FST islands are enriched for these hybridization statistics, like the F statistics and etc. So that's why we think it's an ancestral model. That doesn't exclude that hybridization didn't contribute to FST outlier, right? Because it could be that one SNP with a really high QTL, you know, an important QTL that explains everything could have explained everything. So those are some of the things we need to address in the future.